Hi, everybody. I'm Ben. I'm from the University of Southern California at the Center for Dark Energy Biosphere Investigations. So I have two pieces of bad news. One, I woke up with a really bad head cold today, so I'm being fueled mostly by Dayquil and caffeine. And two, I don't have any phase diagrams in my talk, and I didn't realize that was a requirement for AbSciCon. It's my first meeting, so I apologize for that in advance, too. Just to start, the talk is mostly going to be about phage-host dynamics, and I'm going to make some soft pitches about why evolution is important for thinking about astrobiology, as well as the system that we're working in as an analog for majority of ocean worlds. So even though it's in the title, it will be just a fairly small part of the talk at the end of the day. I want to start off by thanking the people who are part of this project. Most of this work is being done with my graduate student, Elena Graham. I have a lot of conversations with Olivia Nigro about phages. And then there's this big group of people who have all worked on some aspect of North Pond at some point, the site that we work at. And then I'd like to really thank Julie Huber, who brought me on this project a bunch of years ago now and has kept me in the subsurface thinking about these types of processes, even though life moves on and we go different places. So I want to introduce North Pond, which is a site that's off-axis at the Mid-Atlantic Ridge, an 8-million-year-old crust. And this is where I'm going to make my pitch about it being an analog for a majority of ocean worlds. So the ridge flanks that we see off-axis occupy about 70% of the area of the ocean basins. And so while hydrothermal vents are very fun and active and have a lot of energy, it means that a lot of the water-rock interactions that we're thinking about on a global scale are actually happening in colder waters interacting with rocks in these ridge flank areas. North Pond might not be the best example for in-solar system water worlds because it actually has quite a bit of oxygen in it. But if we start thinking about other systems and other solar systems, it might be good to be thinking about these low-energy, cool water-rock interactions that might lead to evidence for life when we get there. And so we can observe, going back the wrong way. We can observe microorganisms in the crust using these Cork observatories, short for Circulation Obviation Retrofit Kits. And so we can hang some equipment off the bottom with this platform here, and we can measure life and look for elements that might be interesting to us as microbiologists in the subsurface. At North Pond, we have two set of corks set up right here. I'm only going to talk about this first one. What we were able to do is we were able to leave these geomicrobe sleds at the top of the well here and sample fluids over a 2-year time course. Represented right here, where we were able to collect DNA and RNA while we weren't there, and then come back and see what that looked like while we weren't there. And so the water comes and flows this way. So we're getting fairly fresh, recharged water. So looks a lot like bottom water, but not quite. And then we'll travel north and discharge on the northeast flank here. And so in the bottom water, we can expect about 10 to the 4th cells per mil. While in the subsurface, we actually get to much lower levels, occasionally, mostly 10 to the 3rd cells per mil. And this is where I want to make the pitch for evolution, right? So hypothetically, we send something somewhere to an ocean world, and maybe we make a really big mistake and we contaminate where we get there, but it's low abundance. And so we're measuring some sample, and we see numbers go up, and we go, we got growth. It's not contamination. It's something that was there from the beginning. But when you're talking about 10 to the 3rd cells per mil, what you're talking about is, if you're a microbiologist, you think 1% is like, okay, 1% of cells. That's pretty good. That's 10 cells. So if you go from 1% to 2%, you're going from 10 to 20 cells. You're really not sure if that's growth, if that's something else that's happening, something that you've contaminated with. So if you add evolution into that, you actually would be able to say, 2 check marks, we have growth, we have evolution. This might actually be something that's a sign of life and something that we can confirm more accurately. So we've answered a number of first-order questions at North Pond, such as which microorganisms are there, what are they doing, and can they shape the chemistry of the marine aquifer? The answer being there are some endemic microbes. So these are microbes that phylogenetically look like they're unique to North Pond. There are also some microbes coming in from the surface ocean, so that would be interesting. That would allow us to ground truth some of our evolution questions. Do we have microbes that aren't really designed to be in the subsurface occurring there? What are they doing? We have evidence to suggest both heterotrophy and autotrophy. And can they shape the aquifer? The answer is yes, through changes in the nitrogen and carbon cycle. If you're interested in this work, there are 2 papers here from Julie's lab that really cover this pretty well. The other thing that's interesting about all this is that when these microbes entered the aquifer, normally through this recharge site, they actually are down there for a really long time. And so this work shows that we can see organic carbon being removed slowly, the longer it's down there for, but that it's actually also aging. So by the time you reach our second site, it's about 2,000 years of time. So less here, so maybe like 200. So we're looking in our 2-year time series about 1% of maybe what could happen over an evolutionary timescale for the consumption of this organic carbon, which allows us to do something because we have time to look at evolution and adaptation. And so we do this by sampling the bulk, cut off here a little bit, sampling the bulk DNA like this. We can use this to then regenerate microbial genomes, which will tell us what they're doing and how many there are of them. And then sometimes, if we're lucky, we can reconstruct phage and viral genomes. So phage are specifically viruses that infect microbes. And so one of the things that happens here is we don't know the difference, or we can't tell who these organisms belong to, right? So the phage are just entities that we have, the microbes are just entities we have. We don't actually know how this interacts, because what normally happens, or one of the classical models of viruses, is that they act as predators. And so here you have a microbe and a phage. The phage infects, kills the host, and then spreads to other organisms. When we look at population dynamics, it looks something like this, where we have the prey, our microbe here being consumed, and then the viruses peaking in abundance afterwards and then collapsing. And this cycle continues as the prey keeps coming up, the viruses then can eat them down, and then it goes on and so forth. But if you're a virus and you're full predator, that's one option for staying alive. The other option is, I made a DNA, you're made a DNA. What if I just hitch a ride with you? And so in this case, the virus infects the host, incorporates itself into the genome, and then spreads by every time the host divides, it becomes part of the host genome. And then there's a mixture of this called a chronic infection, where you can have an infected host that's also producing viruses and also passing it along through its daughter cells. And so I was saying we don't actually know most of the time how viruses and or phages and their hosts are interacting, the microbes, but sometimes we get to do that. We can actually see that using this mechanism called CRISPR, which is actually an innate way for the microbes to prevent infection going forward. And the way this works is when a phage infects a cell, the host will cut it up if it's lucky and doesn't die, and will acquire what is known as a protospacer that matches the virus exactly and incorporates it into its DNA. What happens then is this spacer is expressed, and anytime it sees this virus again, the host, it can actually degrade the DNA before the virus has a chance to infect it fully. So it's a way of preventing infection going forward. And so once we do this, we can actually say this spacer matches this virus, so this host must be infected by this virus. And so we can do that in North Pond, which is pretty exciting. So we can get examples like this, where we have a CRISPR array, where these spacers in green, this one here matches one phage right here. This one has 7 spacers that match one phage. And this one is a fairly small CRISPR that then matches this phage here. And so we know that CRISPR targets are non-random. So this is a recent paper, just came out this year, that shows here in yellow and some of the lighter blue colors that the CRISPR spacers actually target genes in the virus that potentially have some essential part of viral replication. So this is a new element to understanding CRISPR phage and microbe dynamics, because at some point we all thought it didn't matter. If you can just degrade a virus, you can just degrade a virus. Why does it matter what gene you target? But it turns out that some of these genes actually are more important than others, and there's some type of selection potentially happening between hosts and phage, which are selecting for spacers that target certain proteins. So ultimately, can we observe phage evolution that would eventually lead to CRISPR avoidance in our data set? So this means if you're a phage and you're trying to infect a cell and they have your spacer, you're done. You're not getting anywhere evolutionarily. But if you have the ability to change in your spacer, if you can change your DNA just one base pair, all of a sudden you now avoid that CRISPR target and you can go back and be awesome and infect cells as much as you want. So we can track phage abundance and correlate it to host abundance here. So I have in blue in both of these, we have the hosts, and in orange, we have the phage. On this axis, we have just abundance. Phage and host are on the same scale here. Over here, phage are much more abundant relative to the host over time. And then the time are 24 months of sampling that we have. So in this instance, a lot of times the phage correlate exactly directly with the hosts, which is interesting because if you remember the first abundance graph I showed you, there's might be expected to be some offset between where we see the phage and where we see the host, but that could just be the resolution that we're sampling at. And then in some instances, we actually have examples where the host is abundant and we don't see the phage. So this is the meat and potatoes of this talk right here. And so I'm going to walk through this pretty slowly, and then we'll show you 2 more examples of it. So here we have the phage genome all along the x-axis with the genes that represent the phage here. These light gray lines that you can see shifting just represent the coverage, how much of the phage that we can measure in our samples. In this case, time points 1, 3, and 4. So we're having this phage appear and then disappear and then appear and disappear in our samples. And then these large black lines that you can see represent changes at the DNA level. And so these phages here and here and here look different at as you look at the population in these time points than they did and so if you have this nice little cluster here, or this one right here, this one wasn't present here, it's not present here, but it is present there. And then I can overlay where the CRISPR spacer matches and look to see if there's a change in the DNA level of the virus. So in this example, there are 2 CRISPR spacers that hit one particular gene. And in this case, even though we have 6 months of data, the virus doesn't change at all, it's DNA. So the host keeps killing the phage. And at this point, this phage would not be able to infect the one host that we're looking at that we see this match for. It might be able to infect other hosts, which might mean it can still hang around for a little while. But for right now, that's what we show. And it's the second example. We have the CRISPR here, and so we have one sample where this is probably the origin of it because there's no evidence of DNA changes here. But we have one example here where the gene it's targeting is essential for viral replication. And at the same time, there's lots of changes happening on that particular gene, but the CRISPR spacer still hits a region that doesn't change. There's an element of this, which I don't have enough data for, that might suggest that the phage is able to freely allow some mutations to occur on this part of the gene of its gene in an attempt, quote unquote attempt. There's no actual, it's just evolution here, it's not directed. It might actually allow the phage to try and avoid detectants, but it can't. So maybe in the same way that we're selecting certain spacers that hit certain genes for the viruses, the hosts are also selecting certain regions that can't change in the virus. And the last one, the real big daddy of them all. So we have multiple time points on this, again, spacers hitting certain genes, again with annotations that are important to the virus for replication. You have to take my word on it, but for every one of these pink lines, again, it hits a region where there aren't any changes in the DNA, except for this last one right here. In this last one, we actually see changes. So if, in this example, this organism only had one CRISPR spacer, and it was only hitting this one spot, after the first time the phage and host interacted, you would actually have this ability for the phage to keep infecting the host. Unfortunately for it, there's 6 other spacers hitting it, so it's going to keep getting degraded and not be able to infect anymore. So with that, I'll wrap up. So we're able to look at the changes of the phage population at the DNA level over time, that these CRISPR spacers target regions on the phage that seem to not be able to undergo change. If that's intentional or not intentional, we don't know yet. We don't have enough data. And then in all instances except for one, the CRISPRs keep working and the phage dies. So the next step is actually to go back to the host and just look at the host without any elements of phage a part of it. So here's an example where we have an organism that's abundant and then not abundant over time in the green lines. Our reference point actually ends up being our last time point. But as we go back in time, the population of organisms that make up this host actually changes. Its proteome changes. It has changes at the amino acid level in its proteins that might suggest that it's undergoing some type of evolution, either randomly or through some positive selection on there. And if that's the case, then we'd have this change over time, which we could call evolution, and that would give us 2 check marks in terms of both growth and evolution in our samples that we're collecting somewhere else far, far from Earth. And that's it. I'll take some questions.
We have time for one question. Remember, we come up to the microphone. Can you please?
Or just quickly from there.
Yeah, sure.
At the beginning, when you mentioned the possibility of contaminating your system, which is very, very important due to the very low abundance of
Yeah.
salt. Which anti-contamination protocols you are thinking or set in place, if any, or are you thinking to go forward?
Yeah. So the question is about the methodologies you use to sterilize a system going out into space. That's a great question, and I don't have an answer to it.